Hello everyone, welcome back to my channel. Today we are going to study about the very important topic which is protein trafficking. Now, what does that even mean? Think of it like a delivery system inside the cell. When a protein is made, it's not useful until it reaches the correct place, like how a parcel must reach the right address. That whole journey from where the protein is made and where it works, it is what we call it as protein trafficking. Coming to its introduction, so proteins are made by ribosomes, which is most important, must remember this one. Okay, small machines that build them from amino acids. As you all know, proteins are made from amino acids, but the ribosomes does not know where the protein has to go. The cell has a special system that guides each protein to its correct destination inside the nucleus, in the membrane, or even outside the cell. This system is called protein trafficking. And do you know who first discovered this work? George Palade. He used a special technique called pulse chase experiment to track the proteins as they move through the cell. Here you can see the ribosomes making proteins and small arrows showing the journey to different paths. Now, what is so important about protein trafficking? Imagine if a parcel in a city does not deliver to the correct house, it, it reaches to the wrong house. So it creates more complications and complete chaos. If the protein go to the wrong place, the cell cannot function properly. So protein trafficking helps the cell to stay organized. Proteins for secretion go out. Enzymes like liposomes and other proteins stay where they belong. So you can see in the image, each organelle has its own protein destination. That's how neat and organized a cell is. Types of protein transport. There are three main routes for protein transports. First is gated transport, transmembrane transport, and vesicular transport. So let's talk about gated transport. It happens between the nucleus and the cytoplasm through a tiny pores. Second, transmembrane transport, where a protein has to move across the organelle membrane like entering mitochondria. Vesicular transport, when a protein travels inside the bubble and vessels becomes organelles, especially between endoplasmic reticulum and Golgi complex. Look here, you can see arrows showing each type of the pathway. So let's talk detailedly about gated transport, which occurs in the nucleus. So let's start with the first one, gated transport. You know, the nucleus has a double membrane, but there are small holes called nuclear pores. Only the right pore protein can pass through those pores. Like a security gate at the airport, each protein carries a pass. It is called nuclear localization signal if it goes inside, and nuclear export signal which is coming outside. Helper proteins called M protein and X protein checks these passes. This image shows protein entering and leaving through the nuclear pore, like people going in and out of the door with the pass. Now next we will talk about the transmembrane transport. Now imagine proteins that need to go inside mitochondria or chloroplast, they cannot float in it. They must cross the membrane using special channels called translocons. It's like a narrow tunnel. The protein carry a chain and folds properly inside, called chaperones. Protein help guide them so they did not get tangled or misfolded on the way. Here you can see the protein entering and the channel in the membrane that transmembrane transport. Vesicular transport. Third type, the vesicular transport is like a delivery truck in the cell. Proteins are packed in a small vesicles that bug out from one organelle and fuse with the another. For example, proteins move from endoplasmic reticulum to Golgi and then from Golgi to plasma membrane to be secreted. So vesicles are like moving containers carrying the protein safely. See these small round vesicles, they carry proteins between the endoplasmic reticulum, Golgi and the outer membrane. Co-translation. Now, in co-translation, the protein enters the endoplasmic reticulum while it's still being made by ribosomes. A helper particle called SPR recognizes a signal on the protein and takes a ribosome to the endoplasmic reticulum. There, the protein is inserted in a channel called 661 complex and the signal part gets cut off later. This is how secretory and the membrane proteins enter the endoplasmic reticulum. In the picture, see how the ribosome is stuck in endoplasmic reticulum, then why we call it as rough endoplasmic reticulum. Coming to post-translation. In some cases, protein is made completely in cytoplasm first, then post-translation after it is made. It keeps unfolded by chaperones and then pulled into the endoplasmic reticulum using 662 and 663 complex. Inside, another chaperone called BIP uses energy ATP to pull it completely in. Notice the difference here, the translation is already finished before the protein enters. Transmembrane proteins. Some proteins do not go fully inside, they stay attached to the membrane. They have special sequences that decide where they will sit, like whether their head is like face inward or outward. This orientation is decided by some things called positive inside rule. These are receptors, protein, ion channels and transporters. Here you can see how one part of the protein is inside and another part is outside. That's how transmembrane proteins looks. Vesicle codes and directions. Vesicles wear different codes depending upon the direction. So COP2 carries protein forward from endoplasmic reticulum to Golgi. COP1 carries back from Golgi to endoplasmic reticulum, and caltherine move them from Golgi to liposomes or outside during endocystis. Think of these codes as like delivery labels that guide the vesicles. In the image, each type of vesicle is shown in different codes representing its direction. So let's talk about sorting signals. Now, how does the cell know whether to send each protein? It uses short signal sequences like address tags. For example, KDEL for ER, mannose 6 phosphate for liposomes, KSKL for peroxisomes, NLS for nucleosomes. Each of these signals ensure the protein reaches its correct organelle, just like a postal codes. You can see the signals marked. Organelle specific transport. Each organelle has its own entry system. Mitochondria uses TOM and TIM complexes. Chloroplast uses TOC and TIC complex. Peroxisomes uses PST1 or PST2 signals. All of these help the protein enter and settle properly in the right organelle. See the image, the arrow showing the protein entering into the each organelle. That's how specialized the cell transport is. Regulation of transport. Now you might think, how does the cell control all this traffic? It uses small energy molecules called GTPs. For example, RAN GTPs manages transport through the nucleus. and RAB GTPase helps vesicles find their exact destination. They act like a traffic signals, ensuring proteins does not get lost or crashed. Here the small circles labeled RAN, RAB are regulators. They decide where they have to move. Coming to its summary and conclusion, so let's wrap it up. Protein trafficking is like a postal or transport system for the cell. Proteins are made, marked, and transported and delivered exactly where they are needed. This keeps the cell organized and alive. If something goes wrong in this process, serious diseases can be happen. For example, cystic fibrosis. So this is how our cell manages its busy delivery world, all perfectly timed and controlled. Here is the final image shown the full route of ribosomes, organelles and outside of the cell. Thank you for watching my video.